Good evening to all of you. I welcome all of you on the behalf of the Physiological Society of India. And today is the 12th lecture in the lecture series being organized by the Education Committee, Physiological Society of India. This lecture series started in July last year, 2023, and it's almost a year wherein multiple lectures have been delivered on specialty topics. And today is the 12th lecture in this series, which is targeted as undergraduate and postgraduate students. And today we will be talking about the topic of immunology. Today we have two eminent speakers. I have the pleasure to introduce the first speaker, Dr. Kaushik Bharati sir. Dr. Kaushik Bharati sir is the team lead COVID-19 research trackers UNESCO New Delhi and has been a formal consultant for WHO. Sir has obtained his PhD in physiology with a specialization in vaccinology from the Calcutta School of Tropical Medicine and also went on to do postdoctoral fellowship from the Liverpool School of Tropical Medicine UK, where Sir specialized in immunology and molecular biology. Sir has had multiple academic posts, such as Sir was a scientist at the National Institute of Immunology, New Delhi, a senior program officer at the Translational Health and Technology Institute, NCR, New Delhi. Sir was also a visiting professor in the Department of Physiology and Microbiology at Vidyasagar University, Midnapur. Sir has a rich consultancy experience, having consulted for global health strategies, Sanofi Pasteur, France, Policy Curious, Australia, Scientific Advisory Board, Virginia, and Initiative Against Diarrheal and Enteric Diseases in Asia, Lyon, France. The major areas of research by Dr. Bharti sir include virology, vaccinology, and public health. And sir has made tremendous research contributions, whereas he has published 108 papers in reputed national and international journals, and also has co-authored a book. Sir also has served a reviewer for multiple peer-reviewed and scientific journals and is currently on the editorial board of 28 journals across multiple countries. He is editor-in-chief of the South Asian Association of Physiologists Bulletin, Sri Lanka, and chairman of the SAP Research Committee. Sir is also the editor-in-chief of the Journal of Clinical Genetics and Genomics UK, and all the peer review of multiple journals. So now I invite Sir to kindly deliver his talk on the topic immunology and overview. Over to you, sir. Thank you so much.
Thank you for the nice introduction, Dr. Shival. Let me just present my slides.
Yes, sir.
Can you see my slides
Yes, sir.
and my pointer? Let me just make it full screen. Yeah.
Yes, sir. These slides are visible. The stage is all yours, sir. Thank you so much.
Okay, thank you very much. So, as you can see, I'll be talking about an overview of immunology, and we can compare the human body to a machine. Actually, there are many component parts within the body, and they work, simultaneously, just like the cogwheels of a clock. And there are many components, including various systems such as the skeletal system, muscular system, nervous system, uh, cardiovascular system, many systems. So there is much diversity in the body, so many complex parts working together to produce an effect. And but despite this diversity, there is unity as well, as depicted by Leonardo da Vinci's famous Vitruvian Man. So there is also harmony in the body. Many systems, as I just mentioned: digestive system, cardiovascular system, respiratory system, nervous system, excretory system. All these systems, we are all familiar with. But there is another system called the immune system, we are not too so much familiar with. And we can consider this as the invisible army of the body. To put this into context, we must remember that we are all submerged in an ocean of microbes. And these microbes are present everywhere. They're ubiquitous. They're present in the air we breathe, in the water we drink, in the soil, as well as in ourselves, because humans are the most colonized creatures on the planet. And you might be knowing about the gut microbiome, which describes the sum total of all the microbes present within our gut. And these microbes come in different shapes and sizes, and there are four major types of microbes: viruses, bacteria, fungi, and parasites. Now, many of these microbes cause disease. For example, cholera is a diarrheal disease. Malaria, you must all know, it is caused by two major parasites in India: Plasmodium vivax and Plasmodium falciparum, which causes the dreaded cerebral malaria. You also have tuberculosis, which is caused by the tubercle bacillus or Mycobacterium tuberculosis. Of course, you all know about COVID-19 now. It is caused by the severe acute respiratory syndrome, coronavirus 2 or SARS-CoV-2. Then there is also Kala-Azar. This is technically termed as visceral leishmaniasis, and the parasite that causes this disease is called Leishmania donovani. And there are two stages in the life cycle: the promastigotes and the amastigotes. Then we also have dengue. This is a viral disease caused by the dengue virus. There are three types of disease manifestations. The least serious is the dengue fever or DF. More serious is the dengue hemorrhagic fever or DHF. And the most severe is DSS or dengue shock syndrome, which can even kill. Now, it's the immune system's job to keep, these pathogens at bay. The immune system is under constant attack 24/7, you know, three all year round. Besides these pathogens, other things like toxins, carcinogens, pollutants also attack the immune system. So it is being bombarded by all these things day and night. So we can compare the immune system with an umbrella. Just like the umbrella protects us from rain and prevents us from getting wet, similarly, the immune system protects us against the constant onslaught of the various microbes that are thrown against it. So let's have a look at the essential components of the immune system. Looking at the organization, you can see there are several major organs, the major ones being the thymus gland and the bone marrow. Besides this, we have the spleen, we have some, gut associated lymphoid tissue or GALT. These are the Peyer's patches. Besides this, throughout the body, there is an intricate network of vessels called the lymphatic vessels, and these are punctuated with these lymph nodes, which are strategically placed. For example, in the axilla, you have the in the armpits the axillary lymph nodes. In the groin, you have the inguinal lymph nodes. In the neck, you have the cervical lymph nodes. And why I say that these are strategically placed is because whenever a pathogen infects the body and enters the lymph, these lymph drain into these lymph nodes, and the pathogens get entrapped here, and the immune cells, they attack and inactivate these cells, these pathogens. And these lymph nodes, they look resemble a kidney actually, but these are very small, because they have a medulla and they have a cortex like this, and the incoming afferent lymphatics, when there is a infection, the pathogen enters here and they get neutralized within the lymphatic vessels. And these lymphatic vessels are connected with the cardiovascular system throughout the body, as you can see, particularly in the proximal and the dorsal portions here. And these are interlinked by capillaries, so there is free exchange of the various components, cellular components and liquid components between the lymph and the blood. So looking at the cells, all the cells originate from a primordial stem cell. There are two stem cell lines actually, lymphoid stem cell line and the myeloid stem cell line. The myeloid stem cell line produces the major blood cells of the blood vascular system. For example, all the granulocytes, neutrophils, eosinophils, basophils, and mast cells. Besides this, there are the monocytes, including the dendritic cells and the macrophages. In the lymphoid stem cell line, the major players are the lymphocytes. There is a B cell progenitor that produces the B cells and which differentiate into plasma cells, and there are also memory cells. And in the T cell progenitor, it gives rise to T helper cells and cytotoxic T cells. Besides this, there is also the natural killer cells. So let's have a look at some important features of the immune system. One of these features is that immune cells can distinguish between self and non-self. So, our own body cells, they have particular markers. These are called MHC markers, which I'll come to in a moment. And when the cells contain these markers on their surface, the immune cells are pretty happy and they don't attack these cells. So the body is protected. But when a pathogen invades, they have particular markers on their surface called antigen. These are pathogenic determinants, and then it's immune cells become agitated, and so they attack this invading pathogen. So these self molecules that identify our body as being our own, and these are called the major histocompatibility complex. So this term originated from this transplantation reactions, that is when graft is administered from a donor into a recipient, and this can be either accepted or rejected. So this phenomenon of graft rejection or acceptance depended on these molecules called major histocompatibility complex molecules. There are two types, class 1 and class 2. In class 1 MHC molecules, there are three alpha domains: alpha 1, alpha 2, and alpha 3. Alpha 2 and alpha 3 have disulfide bonds, and there is also a beta 2 microglobulin, which helps to stabilize the other three domains. And from the alpha 3 domain, this transmembrane peptide enters into the cytoplasm, and it basically helps to anchor this subunit. And also in this alpha 3 domain, there is a receptor for CD4. CD4 is called cluster of differentiation, and I'll describe this in the next slide. So coming to class 2 molecules, it has two alpha domains, alpha 1 and alpha 2, and two beta domains, beta 1 and beta 2. And these have two transmembrane domains that anchor the MHC class 2 molecule. And in both in the MHC class 1 and in class 2, there is a peptide binding cleft where the antigen binds to these. And here also, just like in the alpha 3 domain, in the beta 2 domain, there is a site, receptor binding site for CD4. So what are these CD8 and CD4? CD stands for cluster of differentiation. So it's a cluster of molecules or proteins that differentiate between different classes of cells. So in a T cell, CD8 marks that these cells will differentiate and generate the cellular arm of the immune response. Whereas the CD4, these mark these cells to become a part of the humoral immune response. So in case of class 1 pathway, what happens is the antigen originates within the cell, intracellular. So how does this happen? Suppose a viral infection has occurred and the virus has entered the cell rather, and within the cell it passes through this machine called proteasome, and these are broken down into peptides, And these are antigenic peptides that pass into the endoplasmic reticulum through tap proteins. These are full form is transporters associated with antigen processing. So, here these antigens bind to the, this is the cleft that I described, in MHC class 1 and class 2. It binds to this cleft, and these are then come outside, in this endosome, and these are presented on the surface of these cells by binding, to the T cell receptors or TCRs in the context of CD8 molecules, which act as co-stimulatory molecules. And then, they stimulate that CD8 T cells and, show these antigens that are being presented to the cell. So, due to exposure to this antigen, these T cells, they differentiate in the pathway that comes next. Whereas in the case of the MHC class 2 pathway, the proteins that enter the antigens, they are extracellular. So they are, endocytosed inside, and it enters the cell. And at the same time, the MHC class 2 molecules present in the endoplasmic reticulum, they are also, secreted out. And here, one thing you should see, that there is an invariant chain that is attached to the peptide binding cleft. And what it does, it essentially protects against non-specific binding by proteins and molecules that may be present in the lumen of the endoplasmic reticulum. Now, when this endosome fuses with this, then this invariant chains dissociates, and the antigen binds to this, and then it is presented to the T cell receptor in the context of CD4 in this case. And when the CD4 cell, recognizes this, antigen, then it goes into the next step of the pathway. Another characteristic feature is that some immune cells, remember pathogens that they have entered the body, and these are called the memory cells. So whenever a immune cell comes in contact with a pathogen, it remembers it. So they can, attack the pathogen if it happens to, infect the cell again. So they are like a police force that patrol the body whenever there's a infection. They will start to activate the immune system. For example, suppose a pathogen infects the cell presents it to the lymphocytes, and it may go into the CD8 pathway, which generates cell mediated immunity, which I'll talk about in a moment. It can also go through a B cell pathway and produce antibodies and neutralize the pathogen. Whereas these cells, these segregated cells, these are the memory cells that patrol the body in case the pathogen happens to enter the body again. I'm talking about this pathogen just as an example. If it enters the body, say, few months later or even a few years later, these memory cells will kick into action. So some cells eat other cells. So phagocytosis, this is called phagocytosis, and it was discovered by the Russian scientist Eli Mechnikov. And it literally means cell eating. So essentially in Greek, phago means devour or to eat, and cyte, as you know, means cell. And he coined the term macrophages to describe these large cells that engulf the infectious agents. And these macrophages are distributed throughout the body. And Eli Mechnikov shared the 1908 Nobel Prize with Paul Ehrlich, Eli Mechnikov for discovering the process of phagocytosis and identifying the macrophages, and Paul Ehrlich for developing his side chain theory of antibody production. So both shared the 1908 Nobel Prize in Physiology or Medicine. So in the phagocyte family, there are many cells, macrophages, of course, that I just talked about. Dendritic cells, these are the antigen presenting cells in the skin. Then we have osteoclasts in combination with osteoblasts also. And osteoclasts belong to the phagocyte family. And both osteoblasts and osteoclasts are used for more remodeling of the bone, whereas osteoblasts build new bone tissue, osteoclasts degrade them. So these are present also. And besides this, you have the granulocytes of the WBCs, monocyte, eosinophils, and neutrophils. So the essential process of phagocytosis, you can see here, it's a receptor-mediated endocytosis, in this case a bacterium, and the endosome or the phagosome, whatever you call it, fuses with the lysosome, and here they get the hydrolytic enzymes present in the lysosomes. They, or the lysozymes, they degrade the cell debris and which are excreted out. And here, this is just a cartoon show to show you that this bacterium is very frightened, because it is being engulfed by a phagocyte. So there are also another class of cells called natural killer cells, which I've termed as the born killers, because they're, inherently they act as killers, and they possess these cytotoxic granules inside. And you can see here two NK cells, one here and one here, which are attacking enemy cell. So the process of killing an enemy cell like this is that the NK cell attaches to the surface. First, it releases these perforins. These are some peptides, and as the name suggests, it creates a perforation on the surface of the enemy cell. And this complex, this hole that is created by these perforin molecules, it's called MAC, or membrane attack complex. Following this, granular enzymes called granzymes, they enter the enemy cell through these, membrane attack complexes, and they start to, devour and degrade the enemy cell, which, essentially it becomes small in size and get engulfed by the macrophage. So this is how the process of killing occurs by NK cells. Let's have a look at the innate and adaptive immunity. So, innate immunity, as the name suggests, is inborn. It is present from birth, and it sets in very quickly. It's very fast because it does not have to depend on antigen recognition. Whatever pathogen enters the body, you know, it becomes activated innate immunity. It's very quick to respond. So it is non-specific. It doesn't depend on the antibody determinant or the antigen present on the surface of these pathogens. Whereas adaptive immunity, this is acquired actually by exposure, repeated exposure to the pathogen. It becomes more specific, so it attacks a particular pathogen, and it is slow because it takes time to adapt to the pathogen in question, and it is dependent on the antigen, that is the surface determinant present on the surface of the pathogen. In case of innate immunity, there are the blood cells, the granulocytes, basophils, eosinophils, neutrophils. There are also the natural killer cells, macrophages, dendritic cells, mast cells. Besides these, there are also complement proteins which are also involved in the innate immunity, which I won't go into today. And in the adaptive immunity, there are B cells and T cells. The B cells produce the antibodies, whereas the T cells differentiate into CD4 and CD8 cells, which are responsible for the cellular immunity. Now, besides these, there are the gamma delta T cells and the natural killer gamma delta cells, T cells, natural killer T cells. And these are components of both the innate immunity as well as the adaptive immunity. So this is just to show you how quick or how time consuming these responses can be. Innate immunity, these are the players, and it takes just a few hours to mount the response. Whereas in adaptive immunity, there is requirement for antigen presentation, and it can take up to days to set in. So if we look at the immune response that is generated in the innate immunity, it is very quick, as you can see, but the intensity is lower. And this is mediated by the IgM antibodies, which I'll talk about in a moment. Then when this declines within a week or so, the adaptive immunity kicks in, and the major players are the antibodies and the T cells. And the antibodies are constituted mainly by the immunoglobulin G. And as soon as the secondary immune response kicks in, the viral load in this case, or it may be any other pathogen, its titer goes down. So this is the essential mechanism. Let's take a closer look at adaptive immunity. So it contains two types of adaptive immunity. One is humoral immunity. This is mediated by antibodies. These are produced by B cells because they are derived from the bone marrow. Since bone marrow begins with the letter B, so these are also called B cells. Then we also have the cell mediated immunity, which is mediated by T cells, and these are derived from the thymus gland. Since thymus begins with T, so these are called T cells, and these directly kill the microbes. So let's take a closer look at the two arms of the adaptive immunity: humoral response and the cellular immune response. Now, when a pathogen infects the body, I've taken the examples of SARS-CoV-2, which causes COVID-19. Now, it may either bind to the BCR, this is the B cell receptor, or it may bind to the TCR or T cell receptor. In the first case, if it binds to the BCR, the B cells, mature into plasma cells, and these plasma cells, eventually neutralize the antigen, which in this case is the spike protein. Whereas in the cellular arm, what happens when the virus attaches to the TCR, the T cells differentiate into two classes. One is the T helper cells or TH cells, and the other is the cytotoxic T cells or TC cells. And these helper cells, as the name suggests, it helps, both the humoral arm by helping to differentiate the B cells into plasma cells, and it also helps the cellular arm for TCs to differentiate into CTLs or cytotoxic T lymphocytes, which directly kill the infected cells. They also secrete some cytokines which help in the killing process. So cytotoxic cells produce toxic agents to kill their targets, whereas helper T cells, they stimulate the B cells to make antibodies, as well as stimulate T cells to become active. So let's have a look at the antigens and antibodies. These are the antigens you can see here, and these are the antibodies in blue. And on the surface of the antigens are these epitopes. These yellow ones, these are the epitopes, and the corresponding binding sites of the antibodies are called paratopes. Okay. Now, when an antibody binds to an antigen, it may be specific or it may be non-specific or cross-reactive. For example, this one, only this triangular paratope is present, which can bind with the triangular epitope. So this is a specific reaction. Whereas in this case, this paratope can bind to this, epitope, but it can also bind to this one. So this is cross-reactive, as well as this. This can bind to here, and this can bind to here also. So this is also cross-reactive. So essentially, what is an antigen? Antigen is an antibody generator. So since the antigen generates antibody, so it is called a antigen. So looking at the antibody structure now, it has a basic Y-shaped structure constituted by a heavy chain here. These two are heavy chains, and two light chains here. This simply means that the molecular weight of the light chains are much more less than that of the heavy chains. And it has a carboxyl domain here at the base, and it has a amino domain at the tips. And they have disulfide bonds here, here, and here also, But this region here, this is called a hinge region, so it allows some degree of flexibility for the antigen binding domain. These are the so-called paratopes that I talked to you about, and these are the various types of antibodies. There are five types essentially, and this IgM, this is a pentamer, five Y-shaped structures you can see here, and this is the active antibody present in the initial primary immune response, that is the innate immune response. Whereas IgG, especially IgG1, class 1 of this IgG, they are the mediators in the secondary immune response. So these antibodies have various other functions, like IgG is involved in allergic reactions. IgG has many functions, and IgA is a dimer. It has a joining chain, J chain, and this is involved in mucosal immunity. So when an antigen and antibody reacts, there is a matrix that is formed, you know, and under certain circumstances, this can precipitate. For example, you can see when an antibody is in excess compared to the antigens, these do not precipitate. In this case also, when the antigen is more than antibodies, here also they do not precipitate. But here, when there is equivalent number of antigens and antibodies, then they precipitate. So the underlying applications for these is that various diagnostic tests use this principle. It's similar types, such as ELISA and other types of diagnostic tools for diagnosing various types of immunological disorders, which my colleague, the second speaker, will talk about. Let's talk about another property. This is called hypersensitivity. This is when the immune system becomes hyperactive and overreacts. Now, there are four types of hypersensitivity reactions. One is type 1 reaction, which is mediated by IgE, and it sets in within 1 hour. One of the major examples is anaphylaxis. Anaphylaxis is a serious allergic reaction, and if epinephrine is not administered immediately, then the patient can even die. And type 2 hypersensitivity reaction is mediated by IgM or IgG, and it takes hours to days to set in. One example is hemolytic anemia. Type 3 is mediated by immune complexes, and it takes 1 to 3 weeks to develop. Common examples include serum sickness and also SLE. This is a common form of autoimmunity called systemic lupus erythematosus. In type 4 hypersensitivity reaction, the mediators are the T cells, and it takes days to weeks to develop. Common examples include rash and SJS. This is Stevens-Johnson syndrome. And coming to the next one, autoimmunity. This is when the immune system can't tolerate self anymore. So I have told you at the very beginning, the immune cells, they can recognize self from non-self. So in autoimmunity, what happens is, the immunotolerance is broken. That is, the immune cells even attack the self cells, so they cannot tolerate. So we face this in everyday life. We have to tolerate a lot of discrimination, criticism, both in the school, bullying, and in the office. So we have to tolerate this, but there is a limit to our tolerance, and when this tolerance breaks, we have an outburst, that is intolerance. And this intolerance occurs exactly what occurs in autoimmunity. So there are many autoimmune diseases that occur, and they affect almost every organ of the body. For example, in the bones you get rheumatoid arthritis and Sjogren's syndrome. In the skin you get psoriasis and dermatomyositis. In the nervous system you can get multiple sclerosis as well as myasthenia gravis, whereas multiple sclerosis affects the brain. Myasthenia gravis affects the neuromuscular junctions and causes flaccidity in the muscles. And the most famous, in fact, just an anecdote, is that the celebrity that is affected by myasthenia gravis is none other than Amitabh Bachchan. So he's affected by this disease. Besides this, the immune system can become affected. Type 1 diabetes mellitus, such as this is also called diabetes insipidus, Graves' disease. In the gut tissue, you can get ulcerative colitis and Crohn's disease also. So I won't go into the details because the second speaker will talk about all this in greater detail. I'll just switch gears and talk a little bit about vaccination. And vaccination harnesses the power of the immune system. So, vaccination is very important because vaccination was discovered way before the discipline of immunology was established. So, if vaccination hadn't been discovered, we might have got immunology much later than we have today. So the story of vaccination originates with variolation, in fact, by this Lady Wortley Montagu. She was an English aristocrat, writer, and poet, and she traveled to the Ottoman Empire in 1716 with her husband. And this is modern day Turkey, and she observed variolation in Constantinople. This is modern day Istanbul, the capital of Turkey. Now, what is variolation? See, when smallpox infects a person, and smallpox was very rampant at that time, and it creates pustules on the skin, on the surface. And in variolation, what is done, these pustules are scraped off and they rubbed into the skin of healthy individuals with the intention of protecting them from smallpox later on. So she was so impressed that she had her son undergo variolation, and when she returned to England 2 years later in 1718, she promoted variolation in England for throughout her life. The actual vaccination story starts with Edward Jenner, who was a simple country doctor who practiced in a small village called Berkeley. Don't mix this up with the Berkeley in California, that is a big city, whereas this is a small village located in the county of Gloucestershire in south of England. You can see it is adjacent to Wales, South Wales, and this is his house. It's called the Chantry, and this is sometimes called the birthplace of vaccination because it is in this house that he first vaccinated James Phipps, who is the first human being to be vaccinated. And the vaccine that Edward Jenner used was a cowpox vaccine. He derived it from cowpox, which are found in milk, and he used this to vaccinate James Phipps. And since this process, is brought about by cowpox virus, cow in Latin means it means vacca, and so since this process uses cowpox, it's called vaccination. So that's how the name vaccination has been derived. And the landmark year was 1796. This is a historical year in the field of vaccinology. Then, about 100 years later, in 1885, Louis Pasteur tried his rabies vaccine in another small boy called Joseph Meister. Here you can see he's being vaccinated and Louis Pasteur is looking on. Interestingly, both James Phipps as well as Joseph Meister, they grew into healthy adults and they did not face any complications arising from vaccination. So this was in 1885. And 2 centuries later, after Edward Jenner did the first vaccination in 1796, in 1980, the WHO declared smallpox to be eradicated from the face of the earth. And this is the first and the only pathogen to be eradicated from the earth. And there's only two samples of smallpox present, one in a Russian lab and one in a US lab, just for archival purposes. And if the need arises, it could be studied. And this is the only one to be eradicated. The next in line is virus, and then followed by measles virus. These are in line for eradication. Now this vaccination process is the same process as in case of infection, but it just mimics the infection. It doesn't it just the antibody antigens are purified, mixed with an adjuvant which boosts the immune response, and the two pathways again occur same: cellular immunity, humoral immunity. So it basically primes the immune system to remain alert and active, so that if the pathogen enters the body, it can become activated and attack the pathogen. So, how effective is vaccination? This diagram shows you clearly how effective it is. Here is the three diseases, dreaded diseases of childhood. In yesteryears: diphtheria, pertussis, this is also called whooping cough, and measles. See the high disease burden initially, but as soon as vaccination was introduced, you can see the cases dwindle so much. So, vaccination is really effective. And just by knowing about the immune system won't suffice. We need to learn to keep our immune system strong and healthy and fighting fit. So, how can we do this? We can do this in the same way as we would do for other systems. For example, we should have a healthy diet that is low in carbohydrates and high in proteins. We have to have meat, fish, and other proteins. Especially, we need to have green leafy vegetables. Drink more water, become remain hydrated. Have seasonal fruits so that it is helpful, for keeping the body active. Lot of antioxidants, lot of vitamins are there. Roughage is there also. And we should avoid junk foods: burgers, french fries, pizzas, all these things, as well as beverages, carbonated beverages. We can also do lifestyle changes, lead an active lifestyle, avoid smoking, get ample sleep, that is also important. Listen to relaxing music, do a little bit of meditation, these helps. And we should also wash our hands with soap and water after using the toilet and before eating. So I mentioned initially that the immune system is always under attack 24/7. So it is already overburdened, and we do not want to overburden it further. So the pathogens that are present in our hands should be cleaned with soap and water, because if we don't wash our hands, then when we eat with our hands, these pathogens will enter the body and overwhelm the immune system. So these are some of the ways that we can not only keep our body holistically healthy, but also the immune system side by side. So with this, I'll stop. I'll just show you, this is a photo of the National Institute of Immunology, and this is the only institute in India that is exclusively dedicated to unraveling the mysteries of the immune system. So thank you very much for your attention.
Thank you so much, sir. It was a very enlightening talk wherein the different aspects of the immune system were discussed right from the basics, the cells, antigen presentation, antibodies, and their functions. And that's a very informative and elaborative talk by sir. Thank you so much, sir. Now I take the pleasure of introducing the next speaker, Dr. Sumi Mukhopadhyay, madam. Madam is presently serving at the Calcutta School of Tropical Medicine, Kolkata, under the Department of Health and Family Welfare, Government of West Bengal, India. Madam has received her doctorate degree from CSIR, Institute of Chemical Biology, Kolkata, and had gathered postdoctoral research experience from Department of Dermatology, Berlin, Germany. The primary area of research is glycoimmunology and infectious diseases, and Madam has published 40 research papers in highly reputed international and national journals. She has made 16 presentations in different symposia and seminars, both national and international. Madam was awarded national scholarship during her graduation and post-graduation and research fellowship by the prestigious Lady Tata Memorial Trust, Department of Science and Technology, and CSIR, Government of India, during her doctoral tenure. Madam was also awarded foreign travel grants from different funding agencies such as CSIR, DST, INSA, Government of India, and she has obtained 11 research grants till date from government sponsors like ICMR, SERV, Department of Science and Technology, DRDO, and many others. Primary area of research where ma'am has contributed a lot is immunodiagnostic of leishmaniasis, biomarker studies in chikungunya and dengue. And ma'am is also serving as an academic editor of the Journal of Tropical Medicine and the Canadian Journal of Infectious Diseases and Medical Microbiology. So today, ma'am will be talking about the disorders of the immune system. So I welcome you, ma'am, to kindly deliver your lecture. Thank you so much.
Thank you. Thank you for that introduction. Am I audible and visible? I'm audible and visible?
Yes, ma'am.
Oh, thank you. So, in the very beginning, I would like to thank the organizers, especially the Education Committee of Physiological Society of India, to offer me this wonderful platform, join you in the PSI online lecture series. The topic of my presentation today is on disorders of the immune system. My previous speaker, Dr. Kaushik Bharati, has wonderfully depicted in a nutshell how our immune system works. So now I'm going to discuss with you how it should work, but it doesn't work. And then we have all sorts of problems in the form of disease manifestations, autoimmune disorders, which I'm going to discuss today. So, in your screen, you find this is already Dr. My previous speaker has already introduced you about the type of immune responses we have. We have the innate immune response and the adaptive immune response, and various cells are involved in this immune system. Uh, the innate immune response are carried out by some specific white blood cells, and the adaptive immune response are carried out by the lymphocytes, primarily the lymphocytes, the T and the B cells. All these cells are produced in the different organs that you find here, the bone marrow, the spleen, the lymph nodes, and they are released into our blood, and they keep a close surveillance and protect us from any type of pathogenic entity, be it a parasite, be it a virus, be it a bacteria. So these are the different cells which have already been introduced to you: the macrophages, the dendrites, dendritic cells, the monocytes, the natural killer cells, the mast cells, the granulocytes, the and also in the adaptive arm you can see the lymphocytes, the B lymphocytes, the T lymphocytes. So what we are going to do, we are going to understand each of these cells and how this particular cell, when they get non-functional or somehow become functions a little bit differently, they manifest in the form of autoimmune disorders. The first cell in your screen are the macrophages. The name, as it suggests, they are primarily they engulf whatever comes in their way. So they are phagocytic in nature. That is, they perform phagocytosis, and primarily they are classified as the M1 macrophage and the M2 macrophage. The M1 macrophages are the ones which are inflammatory. They cause massive amount of inflammation. In fact, these are the ones which immediately rush to any site to rescue us. Then we have the second wave of macrophages, which are the M2 macrophages, which are anti-inflammatory in their function. So M2 macrophages are again subdivided into M2A, B, C, and D. We are not going to into those details, but why I am going to discuss M1 and M2 is because they are very, very important. Their balance are very, very important in different type of autoimmune manifestation. So you can see here the first disease which we are going to discuss today is the systemic lupus erythematosus. It is a chronic autoimmune disorder which is systemic in nature. That is, it occurs throughout our body. It doesn't differentiate or is not organ specific. So you can have it anywhere. The tissue damage can be anywhere, and the culprit is no one but the macrophage. Here you can see the macrophages in SLE. They have very high interferon gene expression, which causes high expression of CD80 or MHC2, that is major histocompatibility 2 expression, which causes more of lymphocytes like T lymphocytes or the B lymphocytes to come and infiltrate in the damaged site. Besides that, these macrophages are dysfunctional. Dysfunctional, that is, they can't perform phagocytosis perfectly. So you have more loading of apoptotic cells here. The cells are not cleared by the system, which the macrophage is supposed to do. And this becomes a source of autoantigen. So these autoantigens are recognized by these the B cells, and they produce huge amount of autoantibodies, which is a characteristic feature of SLE. So you have more of autoantigens, you have more of autoantibody, and then you have huge amount of immune complexes which deposits in various parts like the joint or the skin or the and there they maybe they trigger the complement pathway, or they trigger various types of tissue damaging. They recruit neutrophils, and which causes extensive damage to the affected tissue. And with this, we move on to our next disease, which is the rheumatoid arthritis, which is also a very common autoimmune disorder which we find. But this is organ specific, that is, it mainly affects the joints of our bones. In the synovial fluid, you will find infiltration of all these cells. Primarily, macrophages have a very central role, because these are the cells which send signals in the form of secretion of various type of chemokines. They call these lymphocytes T cells, the B cells, and they also secrete huge amount of IL-26. It is a cytokine which trigger Th17, that is inflammatory pathway, and cause massive synovial membrane destruction, cartilage destruction, bone destruction, which is a very common feature in rheumatoid arthritis. So here also you can see that macrophages have a pivotal role in the manifestation of rheumatoid arthritis. Next disease is systemic sclerosis. This is also, as the name suggests, it is a systemic disease where primarily the skin and the viscera are involved. Here too, macrophages have a very important role. As you can see here, too, phagocytosis is defective. That is, the system is unable to clear apoptotic cells. So this again becomes a rich source of autoantigens, and we have huge amount of secretion of CXCL8, 13, vascular endothelial growth factors, fibronectins, and this all culminates in the formation of tissue damage as seen in systemic sclerosis. Coming to the fourth autoimmune disorder, which is the type 1 diabetes, I think most of us are aware, where the beta cells of the islets of Langerhans of the pancreas are completely destroyed. So macrophages here too plays a very important role. What they do is they trigger the autoimmune cytotoxic T lymphocytes, CD8 T cells, to the site, which keep on destroying the beta cells through their cytotoxic effect. And apart from that, they also recruit neutrophils to the site of damage. And further, macrophages here in type 1 diabetes also hamper the T cell balance, that is the Th1 and Th2 balance. They make the T regulatory cell non-functional. So all this culminates in the manifestation that we see in type 1 diabetes. So to sum up, this table shows you how the macrophages, the polarization of the macrophages can lead to a particular disease manifestation. Like in SLE, we have more of M1 phenotype, M1 macrophage phenotype, and reduced M2 phenotype, which causes increased autophagy of the macrophage. We have inflammatory response, we have reduced phagocytosis. Whereas in rheumatoid arthritis, you can see you have increased M1 phenotype, we have increased anti-apoptosis, which promotes Th17 differentiation, joint inflammation. In systemic sclerosis, on the other hand, we had more of M2 macrophage phenotype and increased anti-apoptosis, reduced phagocytosis to the apoptotic cell, and production of multiple number of cytokines which participate in fibrosis and angiogenesis. Whereas in type 1 diabetes, we have reduced M2 macrophage phenotype, which promotes inflammatory response in the islets, modulate T cell recruitment and activation, and promotes immune cell infiltration and autoimmune response in the islets. The second immune cell which we are going to discuss today is the dendritic cells. The dendritic cells are a very important uh, immune cell because as you can see, the picture itself suggests so many receptors on its cell surface. Besides, it can interact with a number of almost all type of immune cells, be it a cytotoxic T lymphocyte, be it natural killer cell, be it T regulatory cell or B cell. So it has a very central role in our immune system because it trains our lymphocytes, that is the T cells and the B cells, to know what is self and what is non-self. So the tolerogenic capacity of our body, of our immune system, is actually rendered by these dendritic cells. Now, dendritic cells, as my previous speaker has already introduced to all of you, they are produced in the bone marrow. They are produced from the hematopoietic stem cells. They are primarily, again, classified into the classical dendritic cell and the plasmatoid dendritic cell, but the lineages are a bit different. So in inflammatory condition, of course, the dendritic cell phenotype becomes much different. It becomes inflammatory dendritic cell. So dendritic cells have this function of, moving around in the immune system. They nibble around and try to find out anything which is suspicious, anything which is foreign. And whenever they find one, they become activated and they rush to the lymphatics, the lymph nodes, where the lymphocytes are there, the T cells and the B cells are there, so that it can interact, it can present those antigens to the lymphocytes, and those lymphocytes are then trained whether this is self or this is a non-self. So the autoimmunity actually sets in when this tolerance is broken. Dendritic cells have also a very important role in autoimmune disorders. This table shows you how, if you have altered distribution of dendritic cells, that is, if you have reduced dendritic cells, if you have altered or impaired phagocytosis, if you have altered cytokine secretion, the type of cytokine secretion, inappropriate presentation of autoantigens also stimulates dysregulates the dendritic cell function. Besides, there is a defective migration can also cause autoimmune manifestation. So this is a typical picture of how a dendritic cell in an autoimmune disorder like SLE, that is systemic lupus erythematosus, which is, again, I would just tell you that this is a systemic autoimmune disorder where you have huge titers of autoantibodies, you have huge load of autoantigens, and they produce huge amount of immune complexes. Now, these autoantigens are in fact against its own DNA, against its own RNA, again, its anti-nuclear antibodies are there. So these are the immune complexes. So plasmatoid dendritic cells, what they do is they recognize these nuclear autoantigens and they stimulate they stimulate huge amount of type 1 interferons. Now type 1 interferons, they have a very important role. What they do is they block the interaction between the marginalized macrophages and marginal zone B lymphocytes, which are present in the margins of the spleen, that is the lymphoid organ, the secondary lymphoid organ. And you can well imagine, if this is blocked, then the training is incomplete. So the B cells, what they do is they migrate from the margin to the follicle. And this migration or this translocation causes these B cells to be stimulated by the autoantigens, that is the nuclear autoantigens, and they keep on producing, like a feedback loop, they keep on producing high titers of autoantibodies. And so dendritic cells have a pivotal role in this maintenance of this feedback loop in SLE. Similarly, in rheumatoid arthritis also, dendritic cells, as you can see here, they secrete huge amount of IL-17, that is Th17 cells. So you have dysregulated Th17 cell responses, uh, which causes impaired DC migration. So dendritic cells are supposed to pick up an antigen and migrate back to the lymph organs. But here what happens is they stay back. They stay back and they keep on triggering this feedback loop, and this goes on producing huge amount of IL-23, IL-17, and so you have huge amount of Th17 cells, massive inflammation and destruction, tissue destruction. Similarly, in polymyositis and dermatomyositis, which is again autoimmune inflamed muscle conditions, here too you can see that the dendritic cells have an important role. Like in polymyositis, here you can see that they trigger huge amount of CD8 T cell responses. Now, my previous speaker has already introduced how the cytotoxic T lymphocytes, they secrete granzyme B and perforin, which damage these tissues and causing massive tissue destruction. Whereas in dermatomyositis, you can see here that huge amount of auto reactive B cell responses are noted. That is, you have huge amount of autoantibodies. To sum up, dendritic cells appear to be a very important target of therapy in rheumatoid arthritis in SLE, and few therapeutics have completed the phase 1 trials. There are few which, in SLE, where dendritic cell depletion appears to be a novel proposition of therapy, are in the preclinical developmental stage. So dendritic cell-based therapy appears a very important strategy in RA and SLE. Then we are going to talk about the granulocytes. Now, granulocytes, as my previous speaker has already introduced, these are the white blood cells, and they are, especially the neutrophils, are in large number. 50 to 70% of white blood cells that you have are neutrophils, and neutrophils or the basophils or the eosinophils, all of them, as you can see from the picture suggests, they have large number of receptors on their cell surface. They have various number of effector functions, and we are going to discuss one by one and their role in autoimmunity, in rendering autoimmunity. Now, this is a neutrophil life cycle. As you can see, the lifespan of neutrophils are very small, almost a day. They are produced in the bone marrow, they are released in circulation, they follow a circadian pattern, so they are released early in the morning, and by evening they are back to be engulfed by the macrophages. But during this time, they have very important functions, like they can degranulate, they can form net formation, that is neutrophil entrapment, and they can also phagocytose. In fact, these are the cells which come to immediate rescue of any type of invasion, any type of pathogenic invasion that we have in our body. But neutrophils, as I already introduced to you, that they degranulate often, and while they degranulate, they actually cause huge amount of damage to the host also. So if you have more amount of immune complexes, which you find in the kidney glomeruli in the systemic lupus erythematosus, which is there you find that there is more recruitment of neutrophil, and there is reduced net degradation. So if you have reduced net degradation and you have more of complement activation, all this culminates in the formation of tissue damage. Similar, the picture is same in the joints too. You will find that if there is a recruitment of the neutrophils, these neutrophils degranulate and cause a lot of tissue damage. Now, this is a picture of the net traps which the neutrophils set. This is what good neutrophil does to your body. It traps a pathogen. Here you can see a Shigella, the orange colored Shigella, which is trapped by the cell, and it ultimately removes it from your system. So you are saved. But when they are in excess, this is they manifest in the form of autoimmune disorders. The basophils also have very important function. They they actually um identify the autoreactive IgE immune complex, and when they degranulate, they also are not good for the host. So this is a typical picture of that you see in your screen as the role of basophils in the pathogenesis of atopic dermatitis. Here also there is the basophils are activated. When they are activated because of secretion of IL-33 or IL-4 interleukins, they recruit eosinophils, and eosinophils, when they degranulate, they cause allergic inflammation and obviously destruction. uh, Okay, this is a feedback loop which shows in the systemic lupus erythematosus. Here too, you can find that basophils have a very important role, that they keep triggering the B cells to produce autoreactive IgE, and these IgE immune complex in turn keep on triggering the basophil, keep it activated, and this loop, feedback loop, keeps on going in systemic lupus erythematosus. The next granulocyte are the eosinophils. Eosinophils, as you can see in the picture, also have so many granules and so many growth factors and chemokines stored in their granules, and their involvement in autoimmunity is also this table sums up their involvement, like in bullous pemphigoid, which is a very important autoimmune disorder. Here the keratinocytes, the skin cells, they express eotaxin 1, which recruits large number of eosinophils, which degrade the extracellular matrix of the dermal and the epidermal separation. So you have blisters in your skin, and condition is very, very worse. So these are the possible mechanisms by which eosinophil mediated damage in different autoimmune diseases occur. Mast cells also have an important role as far as hypersensitive reactions are concerned. The type 1 allergic manifestations are also brought about by the mast cells. Um, then we have the natural killer cells. The natural killer cells phenotypically are again of two types, the uh, CD56 bright and CD56 dim cells. Both the cells have different functions. Like the dim cells are more in number in the blood, and they have cytotoxic granules, that is perforin and granzyme, whereas the bright cells have more secretion of the cytokines, so they are more immunomodulatory in nature. So if you have a disbalance of this bright and the dim, you can also have a disturbance in your immune system. And this picture in your screen sums up about the role of NK cells in different autoimmune disorders. As you can see in SLE, the peripheral NK cell count decreases, IFN gamma production increases, cytotoxicity is decreased. So you have a high load of apoptotic cells, which again serve as a source of autoantigens, and this loop that we talked about, the autoantibody loop, goes on. So NK cells, I'm not going to discuss each disease here because we have time constraints. So I'll straight away move to the next slide, where you can see the again, the contributory role of natural killer cells in different autoimmune diseases like rheumatoid arthritis or multiple sclerosis or systemic lupus, it plays a very important role, especially recruiting the neutrophils which degranulate and cause tissue destruction. Yes. Then we have a very important component of our immune system, the complement system. The complement pathway is a system of approximately 30 plasma proteins. And these proteins actually recognize any foreign body, and they keep on depositing on the surface, which is known as opsonization. And these opsonized proteins are then recognized by any antigen presenting cell or phagocytes, and they are removed from the system. So a deficiency in any of these components can also be damaging to your immune system. So this is a sum up slide of the different pathways these complement proteins they adopt: the classical pathway, the lectin pathway, the alternate pathway, and various antibodies, monoclonal antibodies or drugs have also been developed also being developed and have been developed to identify that particular notorious component which induces these autoimmune dysfunctions. This is a normal scenario, but in the autoimmune diseases, the pathways are slightly different. Like in SLE, the autoantibodies are against the C1q component of the classical pathway. So if you have a deficient C1q component, you have you have high loading of apoptotic cells, which are again a source of autoantigens and again a source of autoantibodies. So complement deficiency can also trigger autoimmunity. This is a sum up slide of how each component actually contributes to autoimmune diseases. Then coming to the most important part of our immune system, the adaptive immune system, where you have already introduced you the T cell, the B cell, and the killer cell, the CD4 and the CD8 T cells. This is a interaction of the T and the B, how it looks like under the microscope. Now, T cells also have a very important role in mediating, say, skin autoimmunity. There are known four patterns of induction of damage which the T cells cause: pattern 1, pattern 2, pattern 3, and pattern 4. And you can see the cytokine signature in each pattern is a little bit different, and the cascades which follow after that are also a little bit different. I'm not going to discuss in detail because we have time constraints. So this is a very important lymphocyte in as far as autoimmunity is concerned. B lymphocytes, I think by now you have all understood that they are the source of the autoantibodies, so they naturally are very, very important. There are so many receptors on the cell surface, as you can see, and this is how autoimmunity sets in SLE and the important role of B lymphocytes therein. And this is a slide, I think, one of the most important slides, which shows you the therapeutic approaches which have so far been adopted for modulating B cells in autoimmunity. So many drugs have been developed. This is a table which shows you, I think, one of the most well-known molecule is the rituximab, which is targeted against CD20 and approved in rheumatoid arthritis. Similarly, the other molecules also, you can see they are mostly in the phase 1, phase 2, or phase 3 trials. And there is much going on in the B cell lymphocyte therapeutics is concerned. So this is how actually an antigen presenting cell and the T cell and the B cell, they function. They function in coordination. And this is a sum up slide where you can see all the immune cells. They form a network of and they're connected through these different cytokines and chemokines, and a simple imbalance in their secretion or production of a different type of cytokine or chemokine can completely change the scenario, and we have different type of autoimmune manifestation. So autoimmunity has already been introduced to you. So autoimmunity is just not the role of immune cells. Genetics also have an important role. MHC also have an important role. Um, the sex also have an important role. That is, females are more prone to autoimmune disorders, and genes are associated with autoimmune diseases. Um, it is not a singular factor, it is not a single immune cell which causes autoimmunity. There are so many factors, as this table shows you. Um, I will straight away go to, because I think I'm almost done. Um, I'll skip these slides. Okay, this is my lab, and we are actively working on immune cells and biomarkers. And this is the institute where I come from. This is Calcutta School of Tropical Medicine Institute, more than 100 years old, and specifically dedicated to tropical diseases. Uh, Thank you.
Thank you so much, ma'am, for the informative lecture. Ma'am, there are some of the questions which are related to the topics of today in the chat box. So, would you like to take them live, madam?
Yes.
Okay, ma'am. Thank you. I'll read out some of the questions for you. One of the questions is regarding, I believe it has already been discussed to some extent, that is type 2 diabetes mellitus also an autoimmune condition by Dr.
Yes.
Amit Panchal.
Yes. It is also an autoimmune disorder where you have a decrease of the insulin. So it is also another type of, yes.
Okay, ma'am. Then the next question, which is there some way to interpret viral, bacterial, and fungal infections, differentiate between them by using a complete blood count?
Actually, by using complete blood count, you can differentiate viral. In bacterial, you have more of neutrophil. In viral, you have more of NK cells, but it is very non-specific. So at
Okay.
times you may be betrayed. At times the blood picture is, in your favor. So it is. I think it is a very non-specific marker, I would rather say.
Okay, ma'am. Ma'am, then one of the questions by Dr. Srirama, that the M1 and M2 macrophages, they have different clusters of differentiation, ma'am. So is it possible to distinguish between them by CD markers, ma'am?
Yes, yes. Actually, M1 and N2 macrophages can be distinguished by using different CD. I think in my slide, I have shown you the CD markers which help us to distinguish under flow cytometry the M1 and the M2 phenotypes. So we can distinguish them.
Okay, ma'am. Then again, one of the questions by Dr. Srirama is if lung macrophages, if lung fibrosis preventable by inhibiting M1 macrophages, madam?
This, I think, it's under investigation. So this is not my area. So maybe I won't be able
Okay,
to
ma'am.
answer you much, but maybe internet might help.
Okay, ma'am. Okay. Then there is a question by Dr. Mukherjee, that is there any particular age or any precursor health condition for the onset of autoimmune diseases, ma'am?
Actually, yes. Actually, yes. Like endocrinology also has an important role in autoimmune manifestation or age. Like uh, uh, if it is more in females, so menopausal women, they are more prone to autoimmune disorders. So estrogen, the role of estrogen is has a very important role. So age and health condition, yes, they have important role in the onset of autoimmune diseases.
Okay, ma'am. Then one more question is there by Dr. Srirama again. Sir is asking about the role of granulocytes in leukoderma or other skin disorders such as sunburns and depigmentation. I believe this was discussed, ma'am.
Sorry, sunburns or
The the role of granulocytes in depigmentation and other skin disorders.
I'm sorry, I can I have the question again?
I'm sorry, ma'am. I'll repeat it again. Sir is asking whether the granulocytes play any role in skin diseases such as leukoderma, depigmentation, etc. ma'am.
Could be there could be a role, but these are all under investigation. I think every day we are learning more from all the researches that are taking place throughout our world. So one day we will find more number of immune cells also, god knows. So yes,
Okay, ma'am.
could be.
Okay. Thank you so much. Thank you so much, ma'am. Unfortunately, Kaushik sir had another online commitment, so he was not available to take questions. Sir has shared his email. You can share the questions to directed for sir to his email dr.kaushik.bharati@gmail.com. So with this, we wind up today's session on the topic of immunology. Thank you so much, ma'am, for your informative talk once again. And now I would like to invite the President of the Physiological Society of India, Professor Prasunpure Nayak, sir, to deliver the vote of thanks and give further information. Over to you, sir.
Thank you, Dr. Sival. I hope I am audible.
Yes, sir. Yes, sir, you are audible.
Okay. Thank you, Dr. Sival. It's really a nice presentation we had, and I would sincerely like to thank Dr. Kaushik and Dr. Sumi for nice, elaborative, as well as crisp presentations. This online lecture series we have initiated on behalf of PSI by the Education Committee of the Physiological Society of India. It is targeted towards the UG students. So, for the speakers for these lecture series, we have some restrictions where we cannot go into depth of these subjects, and we are requesting the speakers to speak for an hour or so, and in that we are trying to cover as much as possible the subject, and we are taking almost a system, full system which should be taking for, and even for undergraduate teaching, maybe 10 to 12 lectures, and we are trying to cover them within a hour or so. So it's really tough. And this is our 12th lecture series. And we found we have completed roughly all the systems. We have touched upon all the systems, better to say. And now we are thinking to extend these lecture series, which are targeted for the UG student, as I have already mentioned. And we are making it available on the YouTube platform for both synchronous as well as asynchronous learning, so that students in their comfort, they can go through these lectures, they can revise, they can just brush up these lectures. In this context, once we have completed the systems, mostly touched upon the systems, now we are planning to move to a single lecture with specified topics. So next onwards, next session, next July month onwards, next we will be having the fourth Saturday of July, 7 p.m. we will be having lecture. It will be a single lecture, and the topic we have decided, it will be resting membrane potential, and the session will be coordinated by Professor Madhvi Lata. She is from NRI Medical College, member of our education committee, and the topic we have decided it is to be as resting membrane potential, and these topics are initiated with choices by the member PSI education committee members. However, we are requesting the students who are the targeted audience for us to give us, to communicate us with their required topics, so that we can request, we can arrange lectures on those specified topics. However, we do emphasize, we do understand that these YouTube lectures are not to replace the regular classes students are having. This is only to supplement, only to have another option to go through the topics. The July topic, as I have already mentioned, will be on the resting membrane potential, and the topic will be taken, the speaker will be Dr. Padmini, Professor and Head, Osmania Medical College, Hyderabad, Telangana. She will be talking about the resting membrane potential. We are getting good feedback from the students and the teachers, and we are encouraged by your feedback. We are happy that our subscription is increasing and people are liking. I would request all who are attending or who are listening to these classes, they can give their feedback with suggestions, positive suggestions, that what they would like to, and obviously the topics on which we are going to arrange our next classes. Before we end this session, I would like to thank our background team, the PSI Education Committee Coordinator, Dr. Sampa, and the technical part kindly taken care by Dr. Vinay Cheturi from AIMS Rajkot, Assistant Professor from the AIMS Rajkot. Thank you, everyone. And I would also like to extend my thanks to our host tonight, Dr. Sival Srivastav from AIMS Jodhpur, and obviously our both speakers, Dr. Bharati and Dr. Sumi, and also the Physiological Society of India for providing this platform. Thank you, everyone. Hope you have enjoyed these lectures. We'll be